And also, what is important for me there is the part of the absorption, particularly of the lipids. Sure, not only, but still, and as well as the type of co-transport that we have in the case of the absorption of the proteins, particularly amino acids. Sure, I'm talking about, as well as the part of the carbohydrates. Well, as you know, according to on the drive, we do have the presentation for that portion. Um, if I remember, yes, yeah, I do believe you have this presentation. Um, I have gone through it, to be honest, to maybe recheck if I need something here, but I do believe that this part, this like a handout, is really very useful. That when you will go through the guide, it will be very useful to go through it, and will be helpful for you. Well, uh, to start to talk about the digestion and absorption, as you remember in the very beginning of the of our lectures, we said that mainly the important function of the GI tract was to um, provide the nutrients towards the different parts of the body through the blood, sure. And but before that, we know we've talked about some motility portion, yes, and we've talked about some propulsory function of the GI tract. But here, when we talk about the digestion, particularly, mainly, sure, we will talk about the chemical type of the digestion rather than the, for example, mechanical one, which we have already seen in the case of the previous lecture. And here comes what I want you to understand very logical and very simply, because it's not that difficult, first of all. Plus, you already have some basis and some background of the biology information, and you know very well that when we talk about the types of the catabolic and anabolic reactions, when we say about the anabolic and catabolic reactions, where we are talking about the different so like building reactions and vice versa, the degrading reactions, yes. So in this case, there are two types of the reactions which are mainly giving us the simply either degradation or vice versa, the building of particular larger molecules, yes. So in a case of digestion, nothing is changing. Digestion is simply a degradation of a larger molecule into the smaller ones, yes, simply. So as a result, when we talk about the main idea of the digestion of, it will be the any type of a molecule, and in this case, in the case of a digestive tract, we know that mainly the main idea of a food intake to get simply the energy, and we know that three main types, let's like that, of the substances or macromolecules which are providing us with the energy at the end. These are the carbohydrates, proteins, and the lipids. That's why when we talk about the digestion directly, one important thing we need to remember, we do have two types of reactions. Yes, one is condensation type of a reaction, and one is the hydrolysis. So as a result, in the case of the digestion, sure, we are talking about the degradation reactions, yes. So as a result, we are talking about the hydrolysis reaction, and where particularly a larger type of the molecules are simply degraded into the smaller ones, or simply to say into the monomers. So that's why here I brought for you some comparison between the condensation and the hydrolysis. And I do believe it's very clear that when we talk about the condensation reaction, that's what we say about most of the anabolic type of reactions, when we do have the formation of the larger molecule. For example, it will be the polysaccharide, or it can be, I don't know, the polymer of the protein, so polypeptide. Um, Mohini, could you please switch off the mic? Thank you. So, and here we do have the condensation reaction. You can see that very well in this particular picture. Yes, it's okay, Mohini. So here you can see how it looks like. And the main idea here is that two functional groups which are playing the most important role, and these are the hydrogen ion, as you can see here, as well as the part of the hydroxyl one. Yes, so as a result, you can see that in the case of condensation reaction, we always do have the formation of the water as the result. And why? Because from one monomer we have the we are removing the part of the hydrogen ion, as you can see here, and in the case of the other one, we are removing the hydroxyl group. So as a result, we do have the formation of the water. So, and that's what you can see particularly here. But vice versa, when we are talking about the hydrolysis, from the name, it's already clear that we are talking about the cleavage of the water molecule, yes? So as a result, again, we need to, in this case, we are producing the hydrogen and the hydrogen ion, as well as the hydroxyl one. And these ions will be the ones which are helping to, as you can see here, to produce two monomers which are separately formed. Meaning that in case if we do have the large molecule polysaccharide, I don't know, like any other type of a polymer, like polypeptide and so on, the main idea is to cleave the bonding, but the cleavage of the bonding in this case will need us to add the hydrogen and the hydroxyl. Why? Because the formation of that bonding was literally because of the removal of the same type of the groups. As a result, whatever we will talk right now about will be about the idea of the hydrolysis, because the digestion is literally dependent on that type of the reaction, okay? So that's important idea, what you need to remember, and starting to talk about, for example, the digestion of the carbohydrates. What I did, I made the like every and each for every and each substance, I made the digestion and absorption together, so to understand the whole cycle, what happens with particular type of the substance. So, and as a result, here you can see that, first of all, let's talk about, so we will talk about carbohydrates, fats, and the proteins. So starting with the part of the carbohydrates, which is, as you know very well, which is the fast energy source for us, and not only for most of the living things, you can see that mainly what type of the carbohydrates are presented during the food intake, so particularly which are present in the human diet. These are the sucrose, as you can see here, and the sucrose, as you know from the basic biology and chemistry, this is particularly the disaccharide. So the disaccharide, you know, that's simply composed of the two monosaccharides, and sucrose, as you can see, that's what we also call the part of the cane sugar, as well as we do have a part of the lactose, which we know that mainly present in the dairy product. So as a result, in the milk and all of the dairy products, as well as we do have the large amount of the starches, which are polysaccharides simply. And these particular, by the way, yeah, lactose is also disaccharide. That's what I forgot to tell you, that it's also a disaccharide, and we do have a part of the starches, which are polysaccharides. So they're containing a large amount of the different monosaccharides there. But please remember that most abundant, let's like that type of the monosaccharide that we have, this is the glucose. As a result, what we are talking right now about, that from the polysaccharides, how to produce the glucose, as well as we do have the galactose, as you know, as well as we do have the fructose. These are three types of monosaccharides which are ready for the absorption after the complete digestion. So how to come from the polysaccharides towards that portion, that's the most interesting thing, and that's what we'll talk right now about. So starches are important thing. They are coming from the non-animal sources. So mainly we are talking about the, for example, you can see here the potato or any other types of the food. For example, the different types of grains and so on. Important thing that the type of the carbohydrate polysaccharide, sorry, which is sorry, I didn't see the question. Yes, yes, I agree with you. Maltose is also the disaccharide. That's true. So, and what is the most interesting part, that the part of the cellulose which is present, like the simply what we call fiber, yes, in the food, it's also a polysaccharide. It's also a carbohydrate, but important thing that mostly none of our enzymes can digest the cellulose, and as a result, normally it is, but it's still important in the diet. And why? It's mainly important not for the digestion and absorption, but it's important in the formation of the fecal masses, you know. And as a result, that's also true, some facilitation of digestion, but important thing to produce a bulk material of the fecal masses for the normal propagation through the large intestine and its evacuation by the defecation. That's why let's start to talk about particularly the digestion of the carbohydrates. And as we said, mainly what we are talking about here, this is the part of the hydrolysis. That's what you can see here. So during the digestion, yes, so that's what we see. This is the part of the disaccharide. So, and sure, with the presence of the water, that's the idea of the hydrolysis, we do have the formation of the two monosaccharides as the result. So, but now the question is, how, what is digesting the carbohydrates and where they are digested. That's also important. And you can see that there are two places, let's like that, or two parts where we do have the digestion of carbohydrates, and these are the oral cavity, so the part of the mouth, as well as the small intestine, particularly the part of the duodenum. And here you can see that in both of the cases, we do have the amylase as the enzyme. So in the case of the mouth, it's a salivary type of the amylase, and particularly this one is secreted with the saliva, and you will see that in my offline video, how it is secreted, and how with the saliva and all of the physiological part. But what is important, that it is particularly secreted with the help of the parotid glands. So, and as a result, these even sometimes, for example, when you would chew for a little bit longer time and give some more exposure, for example, of the piece of bread or bolus of bread, or some other food types which contain a large amount of the carbohydrates, you can feel that there is some, more sweeten taste. Why? Simply because in this case the digestion of the carbohydrates has started in the oral cavity, and just simply the prolonged chewing will give us the more mechanical digestion, and more important thing, exposure to the saliva and the salivary amylase. So that's why you can see that this is the scheme of how do we digest the part of the carbohydrates. That's why I can see the part of the ptyalin, which is simply the salivary amylase here. And here you can see also that particularly up to 40% of the, it's also sure up to the feeding behavior in this case, because most commonly, as I said, important function of the oral cavity is first of all the mechanical digestion, which ensures the larger surface area to expose to the enzymes. So that's the idea here. That's why most commonly I do believe you have heard that, okay, chew the food for a longer time, it is good. And so sure, it is good, because in this case the digestion will be complete, and as a result you can see that up to 40% of the carbohydrates in this case can be digested by the oral cavity by the salivary amylase. You can see that all of the other percentage of the carbohydrates are simply continued to be digested in the part of the small intestine, particularly the portion of the duodenum, and that's where. But the most interesting part is that simply here comes several type of enzymes which are helping to do that. Okay, we said about salivary amylase, that was clear, but now the second type of amylase, which is the pancreatic amylase, so coming from the pancreas particularly, is giving let's like that, it begins the or continues, let's like that, the digestion. Why I'm saying that? Because important thing, do you see, mostly what we get as the result, this will be just the disaccharides. So the result of digestion by the salivary or the pancreatic amylase at the end is not the monosaccharide which is ready for the absorption. That's what we need to remember. Okay, so as a result, in the most commonly what we have here, this is the type of the maltose, as our colleague, I do believe, Judd, you told us, yes, it's also a type of a disaccharide, and that's true, as well as you can see, 3 to 9 glucose polymers, so containing simply from 3 up to 9 molecules of the glucose, and only at these till this level the amylases can have the digestion. So but it doesn't mean that that's where the digestion is completed. That's not the truth, and particularly that's what you can see here. Back again to the part of the hydrolysis, you can see that this is, for example, on the example of the starch, with the help of the amylase, you can see that we have some oligosaccharides, so with a small amount of the monomers here. We even have the maltriose, for example, with the simply 3 glucoses in the order, or we can have the maltose simply, where we most commonly have two monomers of the glucose, which are still connected to each other and not digested completely. That's why sorry, that's why here you can see that again and again, pancreatic amylase, final product of the digestion by the pancreatic amylase, is a short type of the polysaccharides. So either it's the short oligosaccharides, or these are simply D or trisaccharides, and that's all. Now, you can see that interesting part, why it happens. There is the, how to say, a limit of the pancreatic amylase, limiting of the pancreatic amylase in the case of the short oligosaccharides. As you can see that it cannot hydrolyze the bondings between the glucose molecules in the case of a starch, where we have the branching, because you know that most commonly in the case of the polysaccharides, yes, most commonly they are branch type of the structures. As you can see, for example, here you can see these branchings, and as a result, this is a limitation of the pancreatic amylase. Please remember that part. Although it doesn't mean that there is a limit of the small intestine itself, particularly the part of the duodenum, as well as the portion of even the unum. But what is interesting here is that Now here we do have the enzymes on the microvilli of the epithelial cells of a small intestine. And here, as you can see, it's very simple to remember the names, to be honest, because it's clear that if we talk about the lactose, which is mainly composed of the one galactose and the one glucose molecule, in this case, it is very easy. The type of the microvilli enzyme, this is the lactase, as you can see. In the case of sucrose, which is mainly composed of a part of a fructose and a portion of the glucose, here you can see the part of a sucrase as one of the enzymes, as well as in the case of a maltose, or for example the larger type of the polymers like 3 to 9. In this case, we do have a part of a maltase and the alpha dextrinase, which are helping us to, that's where particularly we do have now the formation of the monosaccharides. That's why we do have a part of a glucose. That's why you can see here. So the glucose part, here is also the glucose part, as well as the portion of the galactose and the part of the fructose. So again and again, here comes the idea that the brush border of our intestines, so where we do have, which is composed mainly of the microvilli, that's why we call it the brush border, that's where we do have these intestinal enzymes needed for the digestion of the disaccharides, as well as the small oligosaccharides. So, and only after that we can have the absorption of the formed monosaccharides. And here comes the most interesting part. As we said, mainly we have 3 types of monosaccharides which will be absorbed. Yes. So as a result, what is the way of the absorption for 3 of them? First of all, as we said, mostly, to be honest, mostly out of all of the monosaccharides which are produced, up to 95% will be still the glucose, okay? Because even in the disaccharides which we talked about, they still contain at least one monosaccharide, which is the glucose. As a result, the amount of the glucose is literally very large. So as a result, the way of the absorption of the glucose, please remember, it needs the active transport for the absorption of the monosaccharides, although there are some exceptions, sure, with the part of the fructose, which I will show you. But here, please remember that you can see that the glucose is, in this case, is mainly transported by the secondary type of the active transport, where we do have the help of the sodium as the co-transport mechanism, and that's what you can see particularly here. Here is the part, so this is the intestinal lumen, here is the part of interstitial fluid, and then towards the portion of the blood, and here you can see itself the epithelial lining of the small intestine. So what is happening here? As soon as we have formation of a glucose, do you see, we need to have the sodium influx. As you can see, the sodium influx towards the intestinal cell, intestinal epithelial cell. And in this case, particularly the influx, that can be dependent on the gradient difference. Yes, difference in the gradient. And as a result, how can we achieve it? So here comes that we do have the active transport, as you can see, on the basal surface, as well as the lateral surfaces of the epithelial cells. We do have the active transport of the sodium from the epithelial cell towards the interstitium. And sorry. So plus, I do believe it's clear why we need here the active transport. As you know very well, the amount of a sodium on the in the part of interstitial fluid, sure, is larger than in the case of the cell itself. As a result, we have the active transport of the sodium ions going out. And as a result, what is happening inside of the cell? So inside of the cell, we do have a lack of a sodium. And as a result, it produces the um, gradient, and by this gradient the sodium from the intestinal lumen is starting to pass towards the epithelium itself, and as a result here the part of the glucose, you can see that glucose is, with the help of the co-transport, is also entering towards the portion of the epithelial cell. So as a result, again and again, when we talk about the absorption of particularly the glucose, you can see that here we need the type of the active transport, okay? But this active transport is dependent on the sodium and the sodium gradient particularly. That's why we call it simply the secondary active transport, because simply that in about the secondary active transport, that in this case we always have two type um, of the substances which are moving, and one literally moves only because of the gradient of the other, but the other is most commonly is the type of the any type of the ion, not any, but for example it can be the sodium as you see in this case, or for example it can be also the hydrogen ion. It's also sometimes produces the part of the active secondary active transport. As a result, you can see that the part of a galactose has literally exact type of the mechanism as, for example, the glucose. So meaning that we still need here the portion of the sodium, and with the help of a sodium we have the entrance toward of the galactose also. But in the case of fructose, that doesn't work like that. So interesting thing that first of all, in the case of a fructose, we mainly have the type of the transport which we call the facilitated diffusion, and plus when through the facilitated diffusion, the fructose is entering towards the epithelium. Okay, I mean the epithelial cells. Yes, that's where particularly we have the phosphorylation of the fructose, and as a result then the conversion of the fructose towards the glucose, so as a result finally what do we have is that the part of the glucose mainly and some portion of the galactose are entering towards the bloodstream. So as a result, what I want you to remember here is that first of all, what type of a transport should we have? So this is the secondary active type of a transport dependent on the sodium, except for the fructose, which is transported towards the epithelial cell with the facilitated diffusion. And by the way, because of that, because there is a facilitated diffusion rather than the active transport, as a result, I do believe it's clear that that the rate of the diffusion of the fructose will be much lower compared to the rate of the absorption of, for example, the glucose and the galactose. So going through the next portion, no, not next portion. So till now, do you have any questions? Guys, can you hear me? No, it was understandable. That's just a second. Just a second, please. Uh okay, Phoebe. Well, but Rani asked me the question if I saw. Yes, yes. So facility. First of all, facilitated diffusion in this case is still dependent on some gradient. Yes, and this is the one thing. The second thing is that when we talk about the active type of a diffusion and active type of transport, and particularly facilitated by the sodium with the large amounts, yes. So sure, the amount of the glucose which will be entering will be still more, and the rate will be faster, even in the case of the galactose, because of the sodium gradient compared to the gradient, for example, of the fructose. As a result, this difference will be seen, and the biggest, let's like that, not the biggest, but in the period of time, the in a period of time, more glucose and more galactose will be for sure passing towards the blood, rather than in the case of the fructose. Although, okay, fructose is still converted to the glucose there, but because of, just in this case, simply having the simple gradient, that's why it will be plus. You see, gradient is also dependent on the amount of the fructose inside of a cell, simply, yes, on both of the sides. So as it starts, okay, maybe at the beginning it will be fast one, but then step by step, because the fructose is entering and it's dependent only on the part of a facilitated diffusion, sure, it will be slower compared to the glucose, which is entering because of presence of a sodium and the high sodium gradient. That's why. So next portion. Again, when we talk, just a second. You're welcome, Rani. Well, the next portion, what do we have here? The part of a proteins or polypeptides and their digestion. And here, to be honest, again the same idea of the hydrolysis. Okay, as I told you, that's why I talked so much about this hydrolysis part and so on, because again the same happens in this case. Okay, so you know very well that the protein structure is mainly simply the amino acidic structure, so composed of the simple amino acids, and here we do have the peptide bonds, and as a result, the same happens here. For the cleavage of the amino acids, in this case, we again need the part of a hydrolysis. So as a result, here you see this picture, which shows you particularly how it looks like the part of for example, the peptide bond formation, but what was literally taken, what part or what groups were taken here. Again, the part of a hydrogen and a hydroxyl. As a result, the hydrolysis will be the answer in this case too. But now, what are the digestive enzymes are helping us to do that? And according to the information which you already have, maybe from the even basic biology, this is simple, that the protein is digestion starts always where? In a case of the stomach. Why stomach particularly? Because that's where we do have the formation of the pepsinogen. As we know, it's an inactive type of the proteolytic enzyme, but normally in the acidic environment, and I do believe it's clear why, approximately at the between somewhere pH 2 and 3, as you can see, the pepsinogen is converted to the pepsin, and as a result, at this pH, it starts the digestion of the proteins. You can see that its inactivation mainly comes on the higher pHs, and particularly here you can see that above 5, the pepsin is simply inactive. So as a result, what is interesting for us is that pepsin, pepsin, we could say, is approximately the exclusive type of the enzyme which can digest the collagen, because first of all, it's in the case of the enzymatic activity, it is really very difficult to digest the part of a collagen. As a result, because we know that in the case when we talk about the proteins, most commonly, sure, we are talking about the type of the food, such as meat, for example, although there are some other proteins, sure, in the case of the in the case of the plant part, but still, when we talk about the proteins in this case, you know that. Sorry, when we talk about the proteins, yes, and we talk about the meat, where particularly we have a large amount of the collagen in the part of the connective tissue of the meat. As a result, important function of the pepsin, although yes, starting the protein digestion, but also particularly the collagen which is present in the food. As a result, what you can see here, this diagram shows you the different levels of the digestion. And the first thing what they tell you, that proteins introduced to the pepsin are digested, and you can see here that approximately 10 to 20% of the total protein, as you can see, is digested, and as a result we have the formation of the polypeptides, which we do have the formation of the proteoses and peptones. The the idea of a polypeptide, I do believe it's clear, yes, that it is mainly composed simply the amino acids, but in the case of proteoses and peptones, this is mainly the terminology which we are using in the case of a digestive system, particularly in the case of the chyme, which is produced, because if from the very beginning they were using these words like a proteoses or peptones as the simply like a proteins, or the digestion products of the proteins, yes, then the idea of a polypeptide was just it's relatively new, let's say like that. So simply in this case, we could say like that the proteoses are a larger products of a digestion of not only the protein, but simply like a meat, for example. It may still contain some part of the lipids. It may still contain some parts of the carbohydrates also, So as a result, less digested structures. These are more proteoses, more digested is the peptones, and the most digested are already the polypeptides. And here comes the idea: as soon as we do have the entrance of the of these structures like the polypeptides or peptones or proteoses towards the small intestine, here comes the same idea as it was in the case of carbohydrates. Again, we have the different types of the pancreatic peptides and pancreatic enzymes, sorry, and these particularly pancreatic enzymes that you see, they are, for example, the trypsin, for example, chymotrypsin, and so on. Or what is also interesting, that these type of these type of the enzymes are again and again, although yes, they are digesting the polypeptides into the smaller structures, but most commonly, again, there is no end product such as the amino acid. So meaning that, again, we need something more additionally, as it was in the case of a carbohydrates and the part of a brush border, as you remember, that will be producing the part of the amino acids. That's why what you see here, all of this pancreatic type of the enzymes are still giving us still smaller type of the polypeptides, some small amounts of the amino acids, but that's not enough for the absorption. And as a result, we do have, again, the formation of the smaller, the formation of the enzymes on the wall of the small intestine, and these are the peptidases. Important thing about, for example, okay, trypsin, chymotrypsin, more or less clear, and we know about that, we heard about that from biology, but what about, for example, the portion of the carboxy polypeptidase? Why it is important for us, or even proelastase? You know very well that first of all, in the case of the even the meat, when we started to talk about the collagen, that's true, it was a part of the connective tissue which is present there. But at the same time, I do believe it is clear that in the part of the connective tissue, we also do have the other fibers, yes, which are the elastic fibers. So the the elastin. So as a result, these fibers should be also digested. In particular, this digestion is mainly dependent on the type of the enzyme, which we call the proelastase. Although the proelastase is also inactive type of the enzyme, and as you can see, entering towards the part of the small intestine, we do have the conversion of the proelastase into the more active and potent type of enzyme, which we call the elastase. And as a result, here we do have the digestion of the of these elastin fibers, and which gives us the opportunity to expose even more surface area to the digestion of the peptidases after. Also, you can see here that as soon as the part of the trypsin and the chymotrypsin, they try to split the large molecules of the proteins towards the smaller one. In this case, now we still have some, as I told you, we still have some formation of the amino acids, and these amino acids are mainly dependent on the part of the carboxyl polypeptidase. Why? Because you know very well, again, from a simple biology or biochemistry, that in the part of amino acid, we do have the carboxyl groups and the carboxyl ends. As a result, from the name of the of this enzyme, you do have you already can understand that we are talking about the we are talking about the digestion and the formation of these small amount, but still of the individual type of the amino acids, which are cleaved from the polypeptides from their carboxyl ends, and as a result can be are ready to be absorbed simply. So that's why we said, although yes, having so many different types of the pancreatic enzymes, it will be trypsin, it will be chymotrypsin, I don't know, it will be carboxy polypeptidase, I don't know, it will be the proelastase, doesn't matter. Still, a large amount of the proteins are not digested completely, even after the pancreatic enzymes, and as a result, we have here again, we have the brush border, and in the very similar as it was in the case of carbohydrates, you see that here we need again the brush border enzymes, which are the peptidases. So, and these are the ones which are either, as you can see, amino polypeptidases. Simply, I do believe it's clear now why it's called like that. If it was carboxy polypeptidases, here you have amino polypeptidases as well as the dipeptidases, which are simply helping us to produce the amino acid as the end product of the digestion, rather than the polypeptides and rather than the small chains of the amino acids. So you can see here that that the part of the amino acids plus the dipeptides plus the tripeptides, as you can see, are very easily transported towards the microvilli membrane, and in this case we do have the part of the peptidases, which are helping us to digest this material, and that's what you can see particularly here in the portion of our epithelial cells in the small intestine. So what is the next part? As soon as we do have the portion of the formation of the amino acids, again, here comes the idea of the secondary active transport. And as a result, again, the sodium is the important. That's why you can see that here from that particular picture, you see that the part of the amino acids are absorbed with the help of the sodium co-transport. So what is important also before the transport itself, you can see that most of the peptide or amino acids themselves, yes, are mainly need to be bound, as you can see, to these transport proteins, and only after that we do have the sodium ion and its gradient, which is helping them to enter towards the epithelial cell. So as a result, coming to the portion of the amino acids and the protein digestion. So again and again, the hydrolysis reaction, several types of the enzymes, several steps. So we've compared to the carbohydrates. Carbohydrate steps were in the part of the oral cavity, then the portion of the duodenum, and then the brush border. In this case, the steps are similar, but starting with the stomach portion, then the part of the small intestine, and then the part of a brush border. Okay, this is for the digestion. For the part of the absorption, it's even easier, because in this case we do have again and again the sodium transport mechanism. Till now, was it clear or not? Good. Now let's have some rest, 10 minutes, please, because it's my third lecture for today. And it's now 18:49. So at 19, so in 11 minutes, let's meet back here. Okay. So let's have some 10 minutes break.